All right, so I'm excited to be here this morning to be able to talk to you all about stuff that God has shown me about viruses over the last decade that I've been doing research. I come from a background of vaccine development with viruses, so I've been doing that for about a decade before God called me here to be a part of Hyde Park, to be in ministry, and it's been a blessing to be here. So let me just open with a word of prayer before I get started. Father, thank you for today. Thank you for creating everything with so much detail. We can see you in everything. God, I praise you for being able to study it, to be able to see it. I just want to pray that you speak through me, that you can show everyone all the amazing things you've done in the world of viruses, in the world of biology. Just pray that you guide me in this talk, that you just open the hearts and the minds of people to hear what it is you want them to hear. Amen. So I wanted to just take y'all through some of the stuff that I've seen over the years. I wanted to start off with a question that a lot of people have. And people always are asking, well, why do we even get sick? Why do we have viruses? Why are they even around? What are we supposed to do with this and come to terms with this? Let's go all the way back to the beginning. In Genesis 1 and 2, all things were created good. Man and woman were created. They were very good. God had purpose in everything that he created. And he seeks fellowship with us. He seeks fellowship with humans, with us, and everything was good until Genesis 3. And then in Genesis 3, we see sin is introduced into the world. Adam and Eve eat fruit from the tree of knowledge of good and evil, and that introduced sin, corruption, and that is something that we have been dealing with ever since. One man brought sin into the world, and now we all deal with the corruption of sin. Now, viruses have very many benefits, but we have to deal with disease and outbreaks now. That is part of what we have to deal with the corruption of sin. And we don't really know if viruses came about before Genesis 3, before the fall, or after. That's something that we don't know. We can see some good. We can see some bad things, some corruption there. But we don't really know where they came about, if God placed them on the planet before or after as a consequence. Here's some things that we do know. Why do we endure illness? One of the reasons, we can go back to disobedience. We see in Judges this phrase over and over again. The people of Israel did what was evil in the sight of the Lord. And then we look at 2 Chronicles 7, something that God has to say to us. When I shut up the heavens so that there is no rain, or command the locusts to devour the land, or send pestilence among my people, if my people who are called by my name humble themselves and pray and seek my face and turn from their wicked ways, then I will hear from heaven and will forgive their sin and heal their land. So we do have punishment for sin there. We see evidence of that, but it's all a call to repentance to bring us back to God. God wants us to turn back to him. But not every illness that we suffer is because we're being punished. Sometimes we're just sick. Sometimes that's all it is. We're just sick. So what do we do when we're just sick? How do we come to terms with that? Why are we just sick? I think one of these things is through trials. God wants to test us, to grow us, to help strengthen us, strengthen our faith. And we know he's always working all things together for good. And Peter talks about this a little bit, where it's like this refiner's fire that we go through to burn out impurities, to strengthen our faith, to grow our faith, to grow our character. And to do that, some suffering is necessary. And if you're thinking about that, thinking, man, I really don't like that concept, I don't want to suffer to get better. Well, may I subject to you that anyone who's gone to an institute of higher education has suffered. We've suffered, and we have gone through some tests, we've gone through some trials. We've had to write some term papers we didn't want to write, pull some all-nighters, but guess what? We paid them to do it to us. So we pay them to do it to us, we go there, but we learn, we grow, we go through that process, and we get better. We come out the other side better. So sometimes we just endure illness to get closer to God, to be strengthened in that. But it's not all bad. God has designed viruses in so many amazing ways that are good. We can see his purpose and his glory in that. And Romans 1:20 tells us, for his invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world and the things that have been made. So we're all without excuse. We can see God in everything, and we have to acknowledge it. So I want to show you today some of the things that I've seen where God has provision in sustaining life. You can see God's glory in the complexity of the virus life cycle, his mercy in our immune systems, purpose and humility through pandemics and outbreaks. These are some things I want to look at today, and I hope you can all see God at work in all of these things. So I want to start out with his provision. And if you don't like germs, I apologize for this slide, but here we go anyway. So there's an estimated 10 to the 31st virus particles on this planet. And just to put that into perspective, we estimate that there's 10 to the 21st stars in the known universe, all the universe. So there's so many viruses on this planet. If we just take a teaspoon of ocean water and analyze it, we can find roughly, on average, 10 to the 7th, 10 million particles just in one teaspoon of ocean water. And even science is starting to figure this out with this paper I have right here. We see phage puppet masters of the marine microbial realm. So a phage is a term for a virus that infects bacteria. And science is starting to see that these viruses in the ocean really control everything that's going on in the ocean. We see purpose in it. Proverbs 16:4 says God has made everything for its purpose. And I love this quote from Tony Goldberg here, an epidemiologist. He said, if all viruses suddenly disappeared, the world would be a wonderful place for about a day and a half, and then we'd all die. All the essential things they do in the world far outweigh the bad things. And let me provide an example here. Plankton, it makes over 50% of the Earth's oxygen. We're here and breathing because plankton is making oxygen. And how do they make oxygen? They need nutrients from bacteria that they don't have access to unless the bacteria die. Viruses are actually killing about 50% of the ocean's bacteria every day to provide these nutrients, to provide us with oxygen. This is just one of many examples that viruses have an impact in our ecosystem, all the good things that they do for us. And let's look at the human gut. Our gut contains over 1,000 bacterial species, and these do so many good things for us. They help in food digestion. They process nutrients, vitamins help with absorption. Medicines that we take, bacteria actually process some of that medicine before we take it in. It helps us with bone density, with formation of blood vessels, nervous system. It protects the lining of our gut. And we have to have a certain amount of bacteria in our gut for all this to function, the microbiome in our gut. And we found over 30,000 unique virus populations within our gut bacteria that help regulate all that. And now there's all of these viruses that do so many good things for us, and we're only aware of about 200 viruses that cause disease in humans. So when we're talking about disease in humans, we're talking about a very small sliver of viruses on this planet that actually do that. And it's just so vast. The world is so vast. So I want to read Job 38:18 to put this into perspective. God is saying, have you comprehended the expanse of the earth? Declare if you know all this. It's only a drop in the bucket, what we know. And we're learning more and more every day, but it's nothing compared to what God has done, things that we'll never know, things that we'll never understand. And we're only just beginning to understand the roles of viruses in all of this. So those are some of the good things that viruses do. I want to now look at some things that we know about viruses. We don't know a lot, but let me share with you what we do know in his complex design of viruses and how this all points to God. It all starts with proteins. We know DNA encodes proteins. DNA is the template for proteins. It tells us when to make certain proteins, how much to make. It tells us everything about protein production. And proteins are life's catalysts. We have proteins in our body that do all sorts of tasks. Each protein has a purpose. Each one was designed to provide things such as structural framework for our cells, for our bodies. Metabolism, movement, communication, everything that we do is powered by the little proteins in our bodies carrying out chemical reactions and processes. They have to have a very specific shape to fulfill their purpose. And I have on the left here an enzyme that's breaking down sugar so that we can process it. These enzymes facilitate the breakdown of sugars. And then on the right, this is an amazing protein. This protein is called kinesin. It actually walks along these little microtubules inside of our cells. So these highways that they travel on are actually a support system for our cells to help them hold their shape. These are called microtubules, and the kinesin proteins actually walk along these to deliver things back and forth within our cells to help the biology process occur. And if you just look at the walking mechanism, this just points to God. This points to this complexity that only he could come up with, how we have these little proteins in our bodies making a walking motion to move things along. And this is where we come up with this concept of irreducible complexity. A design reveals a designer and the attributes of that designer. Whenever things are just so amazing, you look at them and you think, how could this have evolved into something like this? So Hebrews 3:4 says, for every house is built by someone, but the builder of all things is God. So we see his design and all of this complexity in biology, but I want to take it a step further here and look at Hebrews 1:3, talking about Jesus. He is the radiance of the glory of God and the exact imprint of his nature, and he upholds the universe by the word of his power. This is an active thing. God is actively at work through Jesus to uphold the universe. This is not just him sitting back watching. This is him actively doing things to maintain life as it exists. And I want to show you that. So if we just look at how proteins are made, let's just look at how proteins exist, where they come from. It all comes from DNA. And this DNA actually transcribes messenger RNA, which travels into the cytoplasm of the cell. We have ribosomes and tRNA that facilitate the production of proteins. They read this mRNA code, they read this genetic code, and build proteins that do things in our body. These proteins exist as beads on a string initially. They come out as beads on a string. They're this linear molecule, and it goes from this linear molecule and begins to form a secondary structure. One of these is called an alpha helix. There's a couple of different types, but they form this simple structure in the beginning. And they continue to form this more complex structure into a protein that carries out a task. It can carry out an enzymatic task. Multiple proteins can come together to form this larger structure to perform a more complex task. And this is where the amazing thing is. You look at how these fold so intricately, how they come together so intricately from this linear shape to this structure that can carry out a biological process, and you think there has to be something in the cell. There has to be something guiding this process. There has to be all of these things coming together to grab a hold of it, shape it, conform it into what it needs to be. But here's the amazing thing about it. It does it all by itself. Whenever it folds, it just does it. So I just want to say, say you took, the example I could give would be, take a pearl necklace. Pick up a pearl necklace, hold it up in its linear state, and just drop it on the floor. And look at how it lands, that three-dimensional shape that it lands in. Say you did that 10 million times, and it landed in the exact same shape every single time. This is what happens in our bodies. We have millions and millions of proteins, millions of things that just come together just as they're supposed to. All of life, all of biology functions with these proteins. These proteins fold themselves. So I see God at work actively in each and every one of us every second of every day in all of life. Now, to go even further with this, these individual proteins come together to form these complexes that perform very complex tasks. It's not an easy task to replicate DNA, to replicate RNA. Here I have the coronavirus RNA polymerase. Its whole job is to replicate the genome for a coronavirus so that it can pass on its genetic information. It has to attach to the DNA, unfold it, uncoil it, put in the new pieces, the new nucleotides, seal it back up, and essentially what you see here, print out the new genetic material. This is a very complex process that just happens all by itself. These enzymes carry it out each and every day, each and every second in our bodies. So knowing what we do about genetic material and proteins, let's talk about what a virus is. These are the most simple entities in all the planet that can replicate. And you look at them and you're in awe at their simplicity, but at the same time how complex they are. The human body has about 20,000 proteins that we make. This is the influenza virus. This is flu. It only has 11. There's only 11 proteins in it. There are two spike proteins, hemagglutinin and neuraminidase. These two spike proteins are responsible just to get the genetic material into a cell so that it can replicate. We have structural proteins that actually give it its shape and pull everything together to help make the virus particle. Genetic material, which in this case is RNA. Viruses, many of them have RNA, not DNA. Nucleoprotein and polymerase, these carry out the task of just replicating the genome. These are the four essential things that all viruses are going to have, and some of them have non-structural proteins that are immunomodulatory. That's just a way of saying they impact our immune system, to suppress our immune system so that they can replicate and continue to exist. And they all have essentially a similar process with infection. They bind to the cell's surface. The cell actually draws them into a compartment, to a little compartment. And the cell wants to combat the virus initially by doing essentially what our stomachs do. They pump acid into these compartments. They lower the pH to dissolve the virus, to fight against it. But the virus has a mechanism, I'll go into in a minute, to actually let its genetic material out into the cell before this process happens. Once the genetic material gets into the cell, it begins to replicate itself and replicate its proteins. These viruses essentially hijack and take over every one of the cell's processes going on. They shut down everything the cell is trying to do, and all the cell does from that point forward is just make virus particles. Millions of virus particles come out of an infected cell. They go on to infect other cells, and this rapid replication is how viruses continue to exist. So one thing that has baffled us for a while, but we're really starting to figure out and learn more and more detail about how this happens, is the attachment process in getting the genetic material into the cell. It has to form this pore, and these surface proteins actually help that to happen. They actually pinch off the membrane and separate it so that there's a pore that the genetic material can get into the cell through. Now I want to show you some more complexity here. It's some more of God's design here. This is truly amazing how this process happens. This is the hemagglutinin protein in its three-dimensional state here. I have it on the left in the yellow and blue. And I wanted to provide this image on the right as well. So this is colored in a way to show you polarity. This shows you charges. Positive and negative charges, think magnets. Opposites attract. Similar charges repulse one another. So just think about that whenever I go into this process. So these bind to the cell surface, and the cell begins to internalize it. Now, when it gets into these compartments, the pH starts to drop. When the pH drops, it's going to convert some of these charges and flip them. These charges flip, they invert, and all of a sudden, where you have a situation where opposites attract, all of a sudden these components repulse from one another. This is like a loaded spring mechanism that God's designed here. This is a mechanism so whenever the pH drops, these little harpoon-like structures spring out into the cell membrane. And these come from down near the base of the protein where they bind to the virus membrane and invert and come out and stick to the cell membrane. And that continued pH drop as this process happens draws the two membranes together and separate it. So again, irreducible complexity. How does something like this evolve to come about? This is just so complex, how these viruses get in. This is truly amazing. This is something that I've studied for a long time, and I'm just in awe every time I look at it, how this process happens. So when viruses get into a cell, the question that we ask is, how do they survive in increasingly complex organisms? How do they continue to exist? This is where we get into the virus quasi-species. How God has designed these things to work and to survive is a very delicate process, but scientists say they make errors each time they replicate. These are actually directed mutations that the viruses are making. There's some guidance to this. So the changes in the genetic sequence results in diversity within a host over time. As viruses are replicating within us, each and every replication cycle they make, every new cell they go to, they're making little changes in their genetic code each and every time. So over time, what was one virus, one population that gets into you, it starts to diversify a little bit within you. You have multiple virus populations with slightly different genetic sequences, all with different properties. Some of these might be more efficient in what they do. Some of them might be less efficient in what they do. So we have a very diverse population, even in one infection in one individual. Over time, these viruses are searching for efficiency. They're trying to do what they do more and more efficiently as they go. So here, just the image of these peaks, these peaks of efficiency that viruses are trying to achieve. They're quickly adapting to find these peaks. This diversity happens very fast in a matter of days in each and every host. And they're going to operate at these peaks. They don't like to be in the valleys. They want to be efficient. They want to be at these peaks. God has given them a way to get to these peaks. And it's really not a fixed thing. I want y'all to think about this process being dynamic. Each and every host is different. We're all unique. God made each and every one of us unique. Different species are going to have different characteristics. Our immunity is going to play a role in this. But these peaks, these optimal operating zones for viruses are always going to be shifting and changing, and viruses have to be able to change with it. So inside of us and inside of a population, this is what's going on. We start out with a virus that's replicating well inside of us. It might make a change or two that isn't so optimal. Those are quickly weeded out. And then you might have another change happen. So you go from a population of what we have colored here in green, it might shift to red. It might mutate in a certain way that's more efficient, and that more efficient virus starts to overtake the infection. So it's always shifting, always changing, always coming up with new things in new ways. So inside of a host, that process is going on. Say you're going to transmit to your friend on day 5. You're going to cough, you're going to sneeze, they're going to catch the virus from you. The virus that they get is the one most likely that exists in the highest number in your body. So you can see in an individual how it's constantly trying to adapt itself. In between individuals, it's constantly trying to adapt itself. So over time, the most efficient virus is what's going to exist in the population. There's this constant turnover. So why is this important? Why did God have to design it in this way? Well, we have immune systems that are fighting these things. It's this concept of adaptability. There's this perfect balance that has to be maintained. And I say absolutely perfect balance that has to be maintained. A virus can't change so much that those proteins can no longer function. If certain numbers of mutations occur, proteins lose their ability to shape correctly and function correctly. But if they can't change so much that happens, but they have to change just enough to survive in the presence of the immune systems that God has given us. Because if they didn't change, our immune systems would wipe out every virus on this planet. They have to exist. So they have to be able to adapt. So now that we looked at the virus side of things, I want to shift to the immune side of things. We see God's mercy through immunity. God would be completely justified in all of our sin to just allow us to perish at the first infection that we have, but he doesn't do that. Through his mercy, he's given us immune systems. So our immune systems come from our bone marrow. Our bone marrow has a couple of different lineages, the myeloid and lymphoid lineage. The bottom one, the myeloid lineage here, think about first responders. These are the first responders to infection. The reaction isn't all that specific, but it's just to help contain things, to let our lymphoid lineage start to take over. That lineage is our adaptive immune system. It takes time to get the adaptive immune response going to make antibodies, and something has to control it in the meantime. It's really neat how this works together, how God has designed these things to work together. So I'm going to look at influenza again. This is an infection in the lungs. We have these cells called dendritic cells, and we call them that because they have dendrites on them. We're really creative as scientists. We like to come up with creative names. So dendritic cells, dendrites, these cells move along the lung epithelium, those lung cells, and they're sampling constantly to search for signs of infection. These dendrites actually extend out, will sample an infected cell, bring the contents of that cell in to try to identify what's infecting that cell. They internalize these things, and they don't stay there. They move to our lymph nodes, and whenever they get into our lymph nodes, they're going to come into contact with our adaptive immune cells. Now, during this process of moving from the lung epithelium to the lymph nodes, they're doing something. They've internalized these viruses, and in order to make a good antibody, we need to look at fragments of a virus. So these viruses are actually going into these dendritic cells. There are processes in which there are proteins whose whole purpose, whose whole job is to chop up the virus into little bitty pieces. They take these little bitty pieces and present these fragments on the cell surface. They do it in a couple different ways. Through the proteasome, it is essentially like a garbage disposal, just chops up the virus. And then through the acidic compartments that I was talking about previously, it can dissolve the virus into little chunks. Now, the whole reason it does this is so that it can present the immune system with the virus that's infecting it. So this is where the adaptive immunity comes in. We have certain cells that we call T cells. These T cells are going to sample those fragments of virus. They're going to take those fragments to B cells. B cells produce our antibodies. These B cells, through T cells, through dendritic cells, know exactly what antibodies to make to combat the infection. And then we even have a branch of T cells whose responsibility is to go find cells that are infected with that virus and wipe those cells out so they can't spread the infection. So this immune system, this is just on the surface level. There's so many more cells and interactions that take place, but just on the surface level, I want you to just understand the complexity of what God has designed here in each and every one of us to allow us to respond to multiple infections within our lifetime. He gave us the ability to adapt, to make antibodies, to be able to fight off infections. And it's just truly amazing how all of this happens. We always think about antibodies. When we talk about protection, we think about antibodies. And antibodies serve so many purposes. God designed these little proteins can impact a virus in so many different ways. It can in fact impact the spike protein so that it can't function anymore. It can block attachment to the host. It can block processes as it enters the cell so that it can't release its genetic material into the cell. They can impact at multiple points throughout this process. So God has designed a multifunctional protein, one that can adapt to any and every infection that your body encounters throughout your life. And he even provides us with long-term memory. Our memory cells come into play after an infection. We have memory B cells and memory T cells. They hold on to these antibodies that successfully fought off an infection. They keep them stored. And these stay stored so that in the event we come across the same pathogen again, the same virus again, we have an enhanced response that will immediately come into play. It doesn't take us a week to develop the second response. It's already there. It's already ready to go. So that we won't even have symptoms most of the time when we encounter it again. We might have some mild symptoms at best, and that's how he designed our immune systems to work. So this has given us the ability to come up with vaccines. These came about and were pioneered by Edward Jenner back in 1796. He figured out that you could inoculate someone with cowpox and that would protect against smallpox. This is how smallpox came to an end in the world, with cowpox, with a vaccination. And this is before we even knew what viruses looked like. We knew what viruses were. We didn't know what was going on. This is just God allowed Edward Jenner to come up with this concept to spare the world from a continued smallpox plague. And then in the 1950s, we see another successful vaccine campaign. This is really when we started to understand viruses, vaccines, and immunity to some degree. Jonas Salk and Albert Bruce Sabin helped bring an end to polio in the U.S. Now, Sabin came up with this concept of a live attenuated vaccine, a little bit less safe, but a little bit more effective. So if you remember getting those sugar cubes back in the 50s when you went to school, they gave you those sugar cubes, you were getting polio. They were giving you polio. But it was a very mutated form of polio that couldn't go to your brain and cause neurological issues. So this was the concept of the vaccine that was provided then, and they later moved to a killed vaccine once polio was eliminated just to keep it from coming back, something a little bit safer. So through the knowledge that God has granted us in understanding immune systems and viruses, he's allowed us to come up with vaccines to bring eventual ends to bad illnesses circulating. So now I want to combine these concepts with viruses and immunity and talk a little bit about pandemics and outbreaks and how we're really at God's mercy with these things. We think we know a lot, but we really, truly don't. So let's just look at some of the human-transmitted illnesses we're dealing with. We see immune diseases, respiratory illnesses, rhinoviruses, coronaviruses. Those are going to be your common colds. You have seasonal flu, parainfluenza. Everyone knows RSV. If you get a stomach bug, that's a norovirus, most likely, gastroenteritis. Pox diseases, you have hepatitis A, B, and C for the liver. You have some neurological illnesses. God has put viruses on this planet that affects us in all sorts of ways. It affects our organs in all sorts of ways. And it's something that we just have to deal with. Now, here's the area that I studied, zoonotic illnesses. Most of these viruses are lethal in humans, highly pathogenic, and they exist primarily in animals. They exist in the animal kingdom outside of us, but they spill over into humans every now and then. They can cause really severe outbreaks of disease, and we never know when, where, or how they're going to come about. So we have primates, bats, Ebola, you've heard of, Marburg, camels, cattle, rodents. I studied Lassa fever and Argentine hemorrhagic fever, mosquitoes, y'all have heard of yellow fever, Zika, West Nile, all of these illnesses. We never know when these outbreaks are going to happen. And because they exist in animal populations, all it takes is the right genetic change in the virus at the right time and that individual to interact with a human, and a spillover event happens and we have another outbreak. We can't predict it. We try, but we can't. We think we can sometimes, but we truly cannot. This is up to God. This is something that it's too diverse of a population, too diverse of a planet to be able to predict these things. So I just want to show you an example of this with flu. I want you to see just how many species can transmit flu. We have aquatic mammals, bats. We have land mammals, birds. Whenever we have flu events, flu pandemics, these are going to come primarily from birds and from pigs. That's why we had swine flu, we have avian flu, we have all of these different flus that can affect us. And in any pandemic event, these can come from any of these animals really at any time. It's so hard to be able to predict these things happening. We try to intervene. We really do. We try to intervene. But let's look at Job 42:2. I know that you can do all things and that no purpose of yours can be thwarted. So if God wills a pandemic to happen, it's going to happen. It's just going to happen. We try to intervene with interactions with birds, with pigs. We try to prevent interactions, pigs and humans, we try to monitor these interactions to make sure that viruses aren't going to transmit or that we're limiting the chances. But it's too diverse. There's too many animals, too many people. We can't control it. We just can't. We try, and it hasn't been effective yet. And ultimately, it comes down to God is in control of all things. And if he's going to will it to happen, it's going to happen. So that is how pandemics begin, and this is how they sustain themselves. It goes back to this adaptability that I was talking about at the end of the viruses section. There's this adaptability that they have once they get into the population where they're going to change in response to each and everything we try to do. It doesn't matter what we try to do. They have the capacity, God's given them the capacity to change in response to what we're doing. I want to show you one of the complications that we have in that regard with immunity, with vaccines, with anything we try to do, why this is so challenging. If we go back to this hemagglutinin protein that allows the virus to get inside of cells, I want you to remember this mechanism for a second. The orange, the yellow, and the red color-coded part of the protein here on the internal part of the protein, that is that spring mechanism. That's the machine. That's the machine that God's given these virus particles to get into the cell, and they have to be highly conserved, very specific in their sequence, in their shape to do that task. So these areas of the virus don't change much. But the upper portion of it, the head of this protein, the blue part, all it needs is to be able to stick to the cell, and that's highly regulated by this little green chunk that you can see right there on the virus. It's very small. The rest of that blue space has some freedom to change. It can change a little bit in its structure and its shape and still accomplish the task that God's given it. So knowing that, let's look at these antibody binding sites here and see if you notice. Color-coded antibody binding sites, these are the 5 main antibody binding sites that we make antibodies to whenever we have an influenza infection, whether it's swine flu, seasonal flu. They're all at the head of the protein. This is the highly variable portion of the protein that can change at will. So we're making antibodies to a portion of the protein that can constantly change and turn over. It makes our antibodies not very effective. So for one infection, yeah, you have a specific flu strain. You can make antibodies to that flu strain. It's going to prevent viral attachment and membrane fusion, but it's going to be very strain specific. You can get antibodies such as stalk antibodies down here in the conserved region. They're a little bit harder to generate, and they can protect across multiple lineages and types of flu, but they're at times a little bit less effective in preventing infection. So you might have a milder disease, but it's not going to prevent infection. So it's this trade-off. There's no perfect antibody. There's no perfect immunity out there. There's always going to be a little bit of variability in anything we try to do. This is where we get the concept of genetic drift during pandemics. So we have this initial population of viruses that kind of expands into a larger population as it passes through all of us. Our immunity isn't perfect. There's going to be some gaps in it. The virus finds those gaps, circumvents our immune system, and then it leads to a whole new cycle of expansion. We make immunity to that. There's gaps in it. This process goes on and on and on. And we can see that. I'm going to point out part C of the figure here, where we have what we call a phylogenetic tree. This is basically a family tree where you can see the virus adapting over time as it shifts kind of downward at an angle through time here. We look at genetic distance as we move down. We look at time moving left to right. And you can see new strains of viruses popping up over time when you look at this figure and this concept. And with swine flu, that's exactly what we saw. From 2009 to 2019, we look in Kenya. We have lots of samples from Kenya. You can see this stepwise process where you have strain after strain turning over year after year, new strains popping up, old strains being eliminated by the immune system. But our immune systems can never quite wipe it out. It's always here. It's always around. We're always dealing with it. So we have this concept of genetic drift that's going on in things like flu. And then we have another way that past strains can reemerge. Strains that we thought we got rid of long ago, they can always reemerge. As populations die off, as people pass away, their immunity passes away with them. New people are born without immunity. So our herd immunity, as you would call it, disappears over time. Time passes, so let's look at recent pandemics. Take 1918 to 1920, the Spanish flu, one of the worst pandemics we've ever had. This was swine flu. It was H1N1. As the population died off, as the population passed away, it opened the door for swine flu to come back again. We didn't know when or how it was going to happen, but God did. And in 2009, swine flu re-emerged because the population was no longer immune. So this is how we can maintain similar flu viruses throughout the decades, throughout the centuries. Not necessarily this increasing genetic diversity model, but this maintaining model where we see old illnesses coming back time and time again, and once they get into the population, they can shift, they can change, and they can stay. For example, in the 1968-1969 pandemic, H3N2, that's become what we call seasonal flu. It's just stuck around, continued to mutate. Now, there's another concept I want to point out. Respiratory pandemics typically last 12 to 24 months, historically. We're looking at a 1 to 2 year window for respiratory pandemics. And most of that disease burden and death is going to be within the first 12 to 18 months. That's just the cycle of things. That's how God designed these systems so that we would endure it for a while, but that it would eventually come to an end through this relationship between virus and host. So as the host adapts and fights off, the virus actually adapts as well. But with each cycle, the virus gets a little bit weaker and weaker and weaker as it integrates into the population. And this is what we saw in the most recent pandemic that we're all very familiar with COVID-19. So originally, in COVID-19, during the first year, all the original strains were circulating. The virus had adapted to the human population. It was in that adaptation process. There wasn't really widespread immunity yet. Most of the severe disease and death happened right within that first year. And once immunity started to build and the virus started to mutate, then we see that taper off a bit. So as time passed and immunity grew, we see new strains pop up. I'm sure we've all remember from the news the Alpha strain, the Delta strain was the main strain. Then we moved to the Omicron strains. There's just this constant turnover of strains as the virus adapts in the population. And each time we think we have immunity to that strain, it mutates, and we don't have immunity anymore. So these things will persist, just as God said that they would. They tend to persist, and I don't believe there's really any solution to ever prevent this from happening because God has designed all of these things in a way that they can adapt, that they can persist. So as scientists, this is really one of the most humbling things when we look at viruses, when we look at pandemics. We see his intelligent design and how these systems continue to exist, continue to do what they do. And ultimately, we have no control over it. God is in complete control. These pandemics, these outbreaks can emerge anytime from anywhere within the population. They spread rapidly. They cause severe disease and death. And here's the thing, because it's in the first 12 to 18 months typically, vaccine development takes at least 12 months. If you want to be careful, if you want to test the vaccine well, if you want to make sure what you're releasing is a good product that's going to be effective, you need a lot of time to do that. And in order to do that, by the time we get to the point where it's time to release a vaccine, the pandemic has already run its course. So you have to ask yourself, what's the point? If we want to rush the product, if we want to get it out in 3 to 6 months to have any impact at all, it's probably not well tested. It's not something we want to release. But you have to think about this when you look at pandemics and the design that God has for all these things. We're really at God's mercy. If we want to impact the pandemic, it has to be way earlier than we as humans are capable of doing. So ultimately, I hope through this talk you've all seen God's glory and how he designed viruses, how he designed us, designed our immune system, how just incredibly complex these things all are, how it points to a creator who had to create these things. I hope you can see that we as humans ultimately are subject to the will of God. We cannot impact it the way that we think that we can. So that brings us to the question, how do we respond? Well, we have a God that loves us very much, that did give us our immune systems to allow us to persist, to adapt, to be able to grow. He wants us to grow closer to him. That's what he wants. He wants relationship. He's showing us all of these things to show him his power, his majesty. He wants a relationship with him, and he provides us with that through Jesus Christ. We all have sinned. We're all subject to the punishment of sin. We're all subject to suffering, to illness, to everything. But he has given us a way to have a relationship with him through Jesus Christ, who died for our sins, who allows us to have that relationship with him again. So that's what I have for today, and I'm open to questions now.
Test. Ed? Oh, got me. Okay, perfect. So, thank you so much. That was an incredible talk, very informative, inspiring, and to just bring together God's creation, talking about irreducible complexity, certainly something that many of us here in this crowd agree to. So, as you know, John Murphy frequently gets up here at the end of these talks and says, I have 2 rules about the questions that are going to take place to our guest speaker. The first one is ask a question, right? So we don't want you to stand up and give your opinion about something for an extended period of time, but rather ask your question so that we can get on to other questions. And I know there's going to be lots of them. And the second rule, John says, I'm the president, I get to ask the first question, right? Well, he's not here. I'm the vice president, so I'm going to ask the first 2 questions.
All right.
And unfortunately, we are limited with regards to, as you can tell, our microphone system. So once I ask my question and it's answered, I'm going to walk out to the crowd and sort of pick people that have their hands raised and give them the microphone to allow them to ask the question. Everyone can hear it that way, okay? So if you wouldn't mind, maybe comment on gain-of-function research, and particularly with regards to, potentially what that's all about. Is there potential benefit in gain-of-function research? And just some general comments there. And then secondly, with regards to the coronavirus vaccine, if you could comment on how you discuss vaccines. Is our recent coronavirus vaccine different than the vaccines that we used on viruses in the past, and how is that different?
Okay. All right. So Mike's, all right, there it is. All right, still working. Well, it's kind of cutting out.
Ed, are we still good on his?
It's cutting out a little bit. I don't know if.
Turn yours off on the LD.
Okay.
All right, let's try that again. All right, there we go. All right, so question 1. Remind me again. Question. Gain of function. Gain of function. Right. Yeah, that's That's been in the news a lot. Gain of function research. How do we reconcile gain of function research? Really, it's understanding the things in place dealing with gain of function research, why it happens, what we should. Right. Gain of function research. In order to develop vaccines to look at how viruses work, a lot of the time we have the ability to, and one of the strategies is to manipulate the virus genome intentionally. So we're always looking for, if we look at a virus sequence, if we look at what a virus does, the question that we always ask is, what can we change about this virus to make it weaker? That's what we want to do. We want to make it weaker. Our goal is to always weaken the virus. Every now and then, we think we're going to make it weaker. We make a change. We test it. Turns out we made it worse. We made it a lot worse. So this is where the concept of gain of function research came from, is, well, we accidentally made it worse. The general strategy in research is, let's make it weaker, prove that it's weaker, and then go back and make it stronger again back to its original strength. So there's this change that we're trying to make to prove a point, essentially, that something works the way we think it does. Does that make sense? So gain of function research is one of those things that, when done in the right context with the correct oversight and the correct transparency, can be a beneficial thing. But most of the time, it tends to not be done with the correct oversight, the correct the oversight in place and the correct strategy to it, and that's where people can get in trouble with it. With regard to vaccines, the COVID-19 vaccine in particular. So can you remind me again the exact question for that one before I address it?
So I was just wondering if there was a difference between what we saw in the rapid development of the COVID-19 vaccine compared to viral vaccines that were developed in the past. Again, in the news, it seemed to be implied that this is a different type of vaccine for viruses than we have used historically in the past.
Yes. All right. So mRNA vaccines as a concept have been around for about 20 years. They've never really undergone what I would call extensive testing. The mRNA platform is one that, the government really viewed it as this emergency. So they didn't want to take a lot of time to go through a lot of the necessary channels to look at animal experiments. Typically, the FDA wants to see a vaccine perform in a couple of different FDA-approved species before it ever sees people. That process is going to take 6 months to a year by itself. And then you get into a situation where you have clinical studies, phase 1, 2, and 3 clinical trials. You're looking at a year or two for that. So the conundrum there is you've got this limited amount of time to get a vaccine out. It's a new technology. We don't understand what it's going to do in people because it's never seen people before. So when you release the vaccine, are you going to cut some corners and skip a lot of this testing to get it out? Or are you going to take 3 or 4 years to develop it really well, make sure it's all ironed out, and then release it when the pandemic is already gone anyway? So that's the real conundrum that faced them. So we can all see how it played out and what was chosen and what they did. But really, it's just a new platform that hadn't seen people. And anytime that happens, you want to make sure you're careful and you take your time to thoroughly test it to know what you're putting out there. So.
Anyone else have questions?
Question?
I got it. She's closer than you are. Okay.
Thank you so much. I wanted to ask you about mitochondrial function in relationship to viral transmission.
Okay.
The health of the mitochondria, does that impact the transmission of harmful viruses into the cell?
Got it. All right. So mitochondrial function, as we know, mitochondria is essential to making ATP, the energy source for the cell. It is aerobic respiration, as we would call it, is dependent upon oxygen. So mitochondrial health, a healthy mitochondria, again, this is speculation, just speculating here. I don't know that we truly understand everything that goes into it. But speculation would say the more ATP the virus has access to, the more energy the virus has access to, the more copies of itself it can make. So it's really a question of replication efficiency. If it can make more copies of itself, in theory, it's going to spread more readily. But then you have this concept of tissue tropism come into play. So we really don't know how that's going to affect things at a more complex level and how tissue tropism, because you have two questions here. You have the replication aspect of things, making more virus, and then you have the question of infection. Once the virus gets out, how well does it infect? Mitochondrial function would impact, in my opinion, just the first part of it, the replication efficiency. I don't know that there's been any research done on mitochondrial health specifically with regard to virus replication, but it's something I could definitely look into.
This probably dovetails on that previous question. About 3 years ago, Brett Weinstein was asked in a podcast if he felt that coronavirus was man-made. And essentially what he said was, as you said, viruses jump species all the time, but the mechanism for jumping and the mechanism for replication are two entirely different things. So in his opinion, he said, I won't say that it's man-made, but it seems to be man-directed. Would you care to speak to that?
Oh, man. All right. Viruses are very complex. I think to say that any virus is completely man-made would be giving us way too much credit as men. I don't think we understand nearly enough to make something as complex as a virus. That being said, we know that bats harbor thousands upon thousands of coronaviruses. We can go isolate coronaviruses from bats anytime we want. We can find a ton of them. Labs typically collect samples, take those samples to the lab, study those samples. The 2019 coronavirus is unique in that it did have a certain affinity for human lung cells that we hadn't really seen before. So I'll say that. Now, whether that's a lab leak or something that happens to adapt that virus to human lung cells before a leak or anything like that, that's something that we can look around all we want and we're never going to know the answer to. There's things that can point in one direction or another. The fact that it is so adapted to human lung cells was shocking whenever it was released. But any evidence that would point one way or another, I'm sure doesn't even exist anymore.
So this pandemic really took heat on, like a lot of people debating about the vaccines. You know, in our family there was a lot of difference of opinions about should we take vaccines or not. Now many of us have no clue about this, what you just said. You know, we get a little bit of, like, but you being very knowledgeable about this thing, would you mind sharing your opinion about it? Is it okay to take vaccines in general?
Oh, man.
I'm afraid we're going to have to call it.
Oh, man.
So certainly these are difficult questions, right? And I started it. Sorry. Certainly if you want to answer that question generally, and if we want to move on to different subject matter, that'd be great too. Feel free to answer.
All right. Yeah, y'all are bringing the questions today. All right. So, vaccines as a general topic. I'll say this. When a vaccine has gone through all of the necessary testing that the FDA holds a standard to. And I'm not talking about an emergency situation. I'm talking about in general. All of the testing that goes into developing a vaccine, we're talking a decade plus of research to evaluate it in different species, clinical trials. We don't just want to look at healthy individuals, because a lot of the time the shortcut is just to test it in a few healthy individuals and say it's fine. Let's put it out into the population. But you have to consider people that are immunocompromised. You have to consider children. You have to consider pregnancy. There's so many different things that you have to consider with any vaccine. Not one in particular, but any vaccine. You have to consider all these things. And the nature of how these tests happen means it's going to take a decade. You have a lot of data to get through. You have a lot of data to look at before you can stand up in front of people and definitively say that it's okay to take. You're speculating if you haven't put the time in to test it thoroughly. So that being said, it's really an individual risk assessment. It's what do you feel comfortable doing if you don't have the proper data to base a decision on.
The side effects, some people say there's a lot of side effects. Is that true, or any?
So with any vaccine, there's going to be some side effects. The question is, how extensive are those side effects? And if you haven't tested it thoroughly, you're not going to know what those side effects are going to be until it gets into the population. So.
Okay, next question right over here.
Good morning. You were mentioning in the very beginning about the origin of viruses. And my question is, why are scientists or Christians having a hard time understanding the beginning if before the fall, everything was perfect on Earth? Everything that God created was perfect. The animals didn't kill each other. It was paradise. And then the fall happened. Why can't they say that was the dividing line?
So that is a let the microphone kick in here. There we go. That is a good question. Everything was perfect, but at the beginning of my talk, I talked about how viruses have a lot of good functions. And then the question is, God created all living things. Are viruses alive or not? A lot of scientists would say, no, they're not. It's kind of universally accepted that viruses are not alive. They can't reproduce on their own outside of cells. So they're a biological entity that we would consider not living. We don't know how God defines viruses. That's ultimately what matters, but we know how we define them. So based on our definition, the question would be, if they're not living technically, would they have been created before or after the fall? Is that part of our punishment for sin? But viruses do so many good things. I think that's where the hang-up is. Viruses have so many benefits, so we really can't point one way or the other. The Bible doesn't say viruses anywhere, so it's really all just speculation on our part as to how that would have come about, I think.
Question back here. Talk a lot about how viruses mutate so easily.
Louder.
And I read recently that COVID back over lots.
Mm-hmm.
And I'm wondering, would you, in your opinion, professional opinion, attribute that more to how rapidly the virus has mutated so that the vaccines we have are not as effective, or simply complacency within the population of not getting vaccinated?
Right. So with vaccines in general, I want to think about this concept with regard to that question. Vaccines are really more of a roadblock. You want to get out ahead of the infection. You want to get out ahead of the outbreak. And with a virus that's very rapidly mutating, it's very hard to predict where it's going to go next. It's very hard to predict how it's going to mutate. And we know that vaccines take some time to manufacture, so really we're playing a forecasting game with vaccines. We're trying to get out ahead of it and put that roadblock there to curb the spread, but we don't know what the virus is going to look like 3 to 6 months from now when the vaccine is going to be released. So it's guesswork on our part, So really that rapid mutation with regards to vaccines, that's where the challenge comes in. I don't think it's so much vaccine complacency so much as it is we're guessing about what vaccine to make. And we're trying to predict down the road. And you can see annually with the flu vaccine how that's not really a perfect thing that we can do. Every year, the flu vaccine, we're looking at 30 to 60% effectiveness at best because we're guessing the year ahead what it's going to be.
Thank you so much. I have a question about you were talking about the blessing with viruses. In terms of autoimmune diseases, for instance, there are some individuals with Crohn's disease who intentionally ingested like worms so that their immune system would stay busy fighting that rather than their own intestines. My question is, could a benefit of viral infections or pandemics kind of reduce autoimmune diseases over time in the population, or is it more based on the individual?
Right. So that comes down to individual genetics, I think. Where it will benefit some, it won't benefit others. It really depends on how God made us genetically as individuals as to how that's going to impact us. And I'm going to be honest, in terms of viruses, that's not a field where we really have much understanding at all. We look at co-infections. We look at infections in the context of genetic disorders. But it's not something that's been studied extensively enough to really know the answer to that, I don't think. We've only known about the existence of viruses for about 60 or 70 years. And really the field of immunology combined with viruses, that's maybe 20 years old, 30 years old. So we're still learning a lot with regard to that.
Okay, we're going to take 2 more questions. I'm sure my hope is that you'll be willing to stay afterwards for those that you're going to get a crowd up there. So we're going to take 2 more questions and then adjourn. David has a couple of announcements to make after that. So here we go, last 2.
So in the growth of artificial intelligence and machine learning, rapid growth, do you see this as good hope for the detection and producing development of vaccines, or are we limited by the clinical trials? So what's your comment on the role that AI and machine learning can play in the development of a vaccine?
Right. So specifically related to vaccine development and strategies, if we just look within that scope and don't pay attention to anything else going on with AI, just within that scope, it can do a lot of computational processing that we as humans don't have time to sit down and do, nor could we do at that speed. So I think with the right equations, the right inputs, the right rationale, it's going to be able to do a lot that we couldn't and maybe predict some things that we can't. But I still believe that these biological processes and these biological entities as a whole are so complex, I don't think even AIs are going to provide the answers that we're looking for, honestly. Maybe. I really don't think that's going to be the case.
Okay, so gotta ask it. So, how effective are masks in preventing transmission?
Oh man.
He's from your church.
Yeah. All right. I think the best way to answer this is when COVID-19 came out back in 2020. We sat down with environmental health and safety there at UTMB. They gave their input. Everyone gave their input. The decision that we arrived at was if you're going to work on this coronavirus and you're going to do it safely, you need to be in a full head PAPR that prevents any air exchange whatsoever with the outside environment. And that was the recommendation, and anything less than that was deemed unacceptable to prevent potential infection while working with it. So.